Okay, so let's get started. Well, I love helping practitioners connect the dots. So I'm really excited to walk everyone through some of the different ways that gut dysfunction can lead to hormone imbalance. Now, I have a lot of material to cover today, so please forgive me if I talk a little fast. So here are our learning objectives. First, we want to recognize that the gut microbiome can affect hormone balance. Then we want to understand some of the different ways that the gut microbiota can influence hormone balance. We want to learn some microbial signatures associated with hormone-related conditions. And then see how hormones can also affect the gut. So it does go both ways. And then as time allows, a Q&A at the end. Now, of course, many of you are already aware that there are far-reaching effects of the gut microbiome. It's involved in all aspects of health. So the role of the gut microbiome in the etiology and pathogenesis of disease remains one of the top research areas for the past few decades. Evidence suggests that the microbiome has a critical role in maintaining human health in almost every organ system. Imbalances in the gut microbiome have been implicated in a variety of health conditions. Now, today we're going to focus on that hormone gut link. And studies indicate that gut microbes influence estrogen levels in the body and may be involved in the development of hormone-related conditions, including endometriosis and PCOS. The gut microbiota can influence the production and metabolism of hormones. So let's get into it. How the gut microbiome affects hormone balance. Now, I want to start by talking about estrogen excess in the gut microbiome. Now, when we say estrogen excess, we could mean overt high estrogen, which would be an elevated estrogen level, but we may also be talking about estrogen dominance. And this is an increased estrogen level relative to the progesterone level. Now, estrogen dominance may be the result of overproduction of estrogen by the body, changes in estrogen metabolism or excretion, and or low progesterone. And as you can see from this image from the Dutch test, this is an example of both high estrogen and estrogen dominance. Now, symptoms of excess estrogen include breast tenderness, PMS, painful periods, heavy periods, anxiety or depression, fatigue, headaches, and weight gain. Conditions associated with or exacerbated by excess estrogen include endometriosis, uterine fibroids, ovarian cysts, and breast, uterine, and ovarian cancers. So let's talk about the link between the gut and high estrogen. Well, gut dysbiosis can cause high estrogen and estrogen dominance. Now, when I use the word dysbiosis, what I'm meaning is a state that occurs when there is a microbial imbalance. Now, this imbalance can be due to the loss or gain of microbes, or changes in the relative abundance of microbes. Now, there are numerous ways that dysbiosis may cause excess estrogen. The ones I want to focus on today are increased beta-glucuronidase production, increased colonic pH, obesity and increased aromatase expression, and increased histamine production. So let's start out with increased beta-glucuronidase production. It's known that some gut bacteria actually produce the enzyme beta-glucuronidase. Beta-glucuronidase is able to break the bond between estrogen and glucuronic acid. The result of that is that estrogen is released back into circulation instead of being eliminated in the stool. So too much beta-glucuronidase can cause an excess of circulating estrogen. And if it's circulating and going round and not being eliminated, it can build up. Therefore, if there is an overgrowth of bacteria that produce beta-glucuronidase, too much estrogen re-enters circulation, which can lead to high estrogen levels and or estrogen dominance. Now, here's what you may see on a GI map test, which is a stool test, and on a Dutch test, which is a urine test. So in the stool, you can see that there is high beta-glucuronidase activity, and this may manifest then on a urine test like the Dutch, where we see high estradiol. Now, we know the importance of beta-glucuronidase and how this influences estrogen. Here's a great study from 2023. So I just want to highlight a few things from the study. It does say that gut microbial beta-glucuronidase is a key factor in regulating host estrogen metabolism. In fact, they conclude that gut microbial beta-glucuronidase may become a potential biomarker for early diagnosis of estrogen-induced diseases. And regulating that gut microbial beta-glucuronidase activity or transplanting gut microbial beta-glucuronidase producing microbes shows promise for treating estrogen-related diseases. Now, I love this image from this article. Now, at the top, you can see our sources of estrogen in the body. So the ovaries can produce it, the adrenal glands, the adipose tissue, and then that estrogen goes through the liver. In the liver, it undergoes metabolism through phase 1 and phase 2. Now, one particular phase 2 process is called glucuronidation. And so it spits out estrogen in this conjugated form. Now, you can follow that down through the gut, and you can see there are two options. One, that conjugated form can be excreted in the stool, and it's eliminated, it's gone. Now, two, the bacterial beta-glucuronidase can act on that conjugated estrogen and separate it out, and then that deconjugated form can go back into circulation. And then again, if it's circulating, it's building up to higher levels where we could see higher estrogen or estrogen dominance result. Now, this is what we're looking for as we look at a GI map stool test. We're looking for high beta-glucuronidase activity, but we're also looking for high beta-glucuronidase producing bacteria. Now, when it comes to the bacteria, studies have shown that Bacteroidetes and Firmicutes, which are the main phyla dominant within the GI tract, are the primary source of beta-glucuronidases. Numerous species that belong to these two phyla or categories produce beta-glucuronidases. Now, beta-glucuronidases do have varying degrees of activity. There is some thought that the beta-glucuronidases produced by bacteria in the Firmicutes phylum may have a higher degree of estrogen reactivation than those produced by bacteria in the Bacteroidetes phylum. But remember, gut microbial beta-glucuronidases modulate the level of systemic estrogen. Okay, now let's look at the next way that dysbiosis may cause excess estrogen. The second way is increased colonic pH. Now, we want a more acidic colonic pH or a low pH. Short-chain fatty acids produced by commensal bacteria in the gut ensure that the colonic pH stays on the acidic side. But if there are low levels of short-chain fatty acid producing bacteria in the colon, the pH rises. Now, where this is important is beta-glucuronidase is an enzyme whose activity is increased at a higher pH. Therefore, increased colonic pH can lead to increased beta-glucuronidase activity. And that's what the GI map is actually measuring. The marker beta-glucuronidase on the GI map represents beta-glucuronidase activity. And the issue is that increased beta-glucuronidase activity causes increased recirculation of estrogen. So what we're looking for on the GI map, we're looking for decreased levels of commensal bacteria that make those short-chain fatty acids. So you may see low commensal or keystone bacteria, or what we call an insufficiency dysbiosis pattern. Now, commensal bacteria consume fiber for the most part. And when they eat that fiber, they ferment it and produce short-chain fatty acids. And it's these short-chain fatty acids, as the name suggests, acids, help acidify the colon. So what this means is a higher colonic pH is going to result in increased beta-glucuronidase activity. And a lower colonic pH, what we want and what we should have, means there could be decreased beta-glucuronidase activity. Now, it's interesting to note that a low colonic pH can both inhibit the growth of some beta-glucuronidase producing species and make them behave better. So for instance, Bacteroides species don't overgrow or produce as much beta-glucuronidase when the colonic pH is lower. So again, pH is very important. Okay, let's move on to another way that dysbiosis may cause excess estrogen. Again, studies have shown that dysbiosis has been linked to obesity. Now, the thing about obesity is it can upregulate the activity of a different enzyme called aromatase. And aromatase converts the androgens androstenedione and testosterone into the estrogens. So E1, which is estrone, and E2, which is estradiol. And the issue is that increased aromatase activity leads to increased aromatization of androgens to estrogens, resulting in excessive levels of circulating estrogens. Now, here's again another image from the Dutch test. And you can see there are a couple arrows that head down from testosterone and androstenedione into the estrogens. So again, we don't want upregulated activity of this enzyme aromatase, or we can end up with too much estrogen. So those are those two enzymes there. You can see the aromatase. Now, talking about obesity, here are some of the studies that link the dysbiosis phenomenon with obesity. Here's a great article from 2021. Dysbiosis of the microbiota causes metabolic disorders and promotes the occurrence and development of obesity. The gut microbiota affects appetite, energy absorption, fat storage, circadian rhythm, and chronic inflammation, leading to obesity. Again, their conclusion, dysbiosis of the gut microbiota has been shown to be closely linked to obesity. They also point out that many gut microorganisms have been identified to be related to obesity. Now, when it comes to organisms related to obesity, there are a few that I did want to highlight. First, let's talk about Akkermansia and the butyrate-producing microbes. Now, these have anti-obesity effects, so we want Akkermansia and these butyrate producers. Having decreased levels of Akkermansia muciniphila and butyrate-producing bacteria can promote obesity. And here's why, at least one of the reasons why. They actually stimulate the secretion of glucagon-like peptide 1, GLP-1, all in the news today, and peptide YY, PYY. These are hormones that have anti-obesity effects. So Akkermansia, what it does is it produces a protein called P9, which stimulates L cells in the intestine to produce GLP-1. So we talk about the GLP-1 agonists and the injections and so forth. Well, your gut, if it's healthy, actually should be producing GLP-1. But we need the right microbes to do that. Now, Akkermansia also produces the short-chain fatty acid propionate, which stimulates the release of both GLP-1 and PYY. And then those butyrate producers, well, they eat fiber, they produce butyrate, and then that butyrate induces a secretion of GLP-1 and PYY. So on the GI map, what we're looking for here is, do we have low Akkermansia? Or do we have low butyrate producers? Now, the butyrate producers on the report we're going to focus on are Faecalibacterium prosnitzii, Roseburia species, and then the Firmicutes phylum. Now, Firmicutes is a large category, but most butyrate producers do fall within that category. So that's what we're looking for on the report. Now, there are some bad guys that have more obesity promoting effects. So let's talk about them. We might have high hydrogen sulfide producers in the gut. Now, this is an interesting study, brand new, August of 2024. They found that Disulfovibrio, which is a bacterium that can live in the gut, so this is capable of producing hydrogen sulfide. And the hydrogen sulfide produced by this organism was found to inhibit the mitochondrial respiration and induce the unfolded protein response in intestinal L cells, thereby hindering GLP-1 secretion and gene expression. So the conclusion is that Disulfovibrio-derived hydrogen sulfide compromises GLP-1 production. Now, what we're looking for on the GI map, yes, we do have Disulfovibrio on here. We can certainly look for that, but it's not the only hydrogen sulfide producer. There's actually quite a few organisms in the gut that are capable of producing hydrogen sulfide. So here's a list of some of the primary hydrogen sulfide producers. So again, keep in mind that studies do now indicate that hydrogen sulfide damages the mitochondria of these GLP-1 producing cells, and that resulting GLP-1 deficiency can then drive insulin resistance, which can lead to obesity. So here's another way. So hydrogen sulfide, again, too much is a bad thing when it comes to obesity, but we need to look at another thing that we can also get from the gut microbiota, which is LPS. LPS is lipopolysaccharide. We don't want too many lipopolysaccharide producers in the gut. So they do show in this study, which is from 2019, that it's thought that one of the mechanisms by which the gut microbiota could contribute to the pathogenesis of obesity and the related metabolic diseases include increased intestinal permeability and bacterial lipopolysaccharides. So again, we're looking to see on the report, do we have high levels of some of these LPS producers? Now, LPS producing bacteria are gram-negative bacteria. So this LPS is part of the outer cell membrane of all gram-negative bacteria. So here's a list of these organisms that have that LPS in their outer cell membrane that you can find on the GI map. And this is a report that we might see that has some high levels of those LPS producers. Okay, now let's look at another way, and the final way that we're going to discuss today on how dysbiosis may cause excess estrogen. And this way is through increased histamine production. We know that some microbes can produce histamine either directly or they stimulate mast cells, which then release histamine. The issue is that then this histamine can stimulate the ovaries to make more estrogen. And this can create a vicious cycle. So you have the estrogen stimulating the mast cells to release more histamine. And then the estrogen can actually downregulate the DAO enzyme that clears histamine. So what we're looking for on the GI map is increased levels of these histamine producers or mast cell activators. And here's a list of those bacteria to look for on the GI map report. Okay, so we talked about estrogen excess in the gut microbiome. Let's look at the opposite side of the equation and look at estrogen deficiency in the gut microbiome. There is actually some correlation there too. First, let's talk about low commensal bacteria. Now, we talked about high beta-glucuronidase. Well, actually, low beta-glucuronidase could be an issue for the opposite reason. A lack of normal beta-glucuronidase producing bacteria can lead to low beta-glucuronidase activity. And the thing is, especially like postmenopausal women, we do rely on having some beta-glucuronidase to recirculate some of that estrogen, especially if we're not producing as much. So low beta-glucuronidase activity means less estrogen is recycled, and this can lead to reduced estrogen levels in the body. So if you see a picture like this in the gut where all these commensals are low or less than DL, you might actually see that there's not enough normal beta-glucuronidase producing bacteria to have a normal beta-glucuronidase activity level. So again, there's a sweet spot. We don't want it too high or too low. When it comes to the gut microbiome, it really is about balance. Now, there's also some new research showing that there are some organisms that actually degrade estrogen. So what if you have high estrogen degrading microbes? So there is some research that some Klebsiella aerogenes can degrade estrogen. And they've actually linked this with a decline in estrogen and depression. So here's a great study here from 2023. So last year. So it's saying the prevalence of this Klebsiella aerogenes and the 3-beta-HSD was higher in premenopausal women with depression. So again, we need a balance of these good commensal bacteria so we don't have too high beta-glucuronidase activity, too low beta-glucuronidase activity, too much estrogen, or too little estrogen. Okay, let's move on to another topic. How we metabolize our estrogen. So let's talk about how the gut microbiome can lead to poor estrogen metabolism. The gut microbiome can actually affect all three phases of estrogen metabolism. So estrogen is metabolized in the liver in two main phases. Phase 1, which is hydroxylation, and phase 2, which includes methylation, sulfation, and glucuronidation. Now, many nutrients are required for phase 1 and phase 2 if there is a lack of these cofactors. So B vitamins, minerals, amino acids. There's impaired function of phase 1 and phase 2 estrogen detoxification. Now, where does the gut come in here? Well, the health of the gut affects the absorption of these nutrients. If your gut isn't healthy, you may end up deficient in these nutrients, and then phase 1 and phase 2 just can't work properly. Then we also know that enzymes involved in phase 1 can be affected by the gut. So 16-OH or 16-hydroxy metabolites are produced by the enzyme CYP3A4. Well, CYP3A4 is present not only in the liver, but also in the small intestine. Inflammation in the gut can upregulate CYP3A4, leading to a higher level of 16-hydroxy metabolites. So it's important when you are looking at, say, a Dutch test, you see this kind of picture where it's just that pie graph is just overwhelmed in blue. 16 hydroxys, in this case, 75%. We don't want that over 30%. We need to be thinking when we see that, is there gut inflammation? Let's look at a stool test and see if there's some pathogens, some dysbiosis, something going on in the gut. Because gut inflammation is one of the top factors that can drive estrogen metabolism down this 16-hydroxy pathway. So that's phase 1 and phase 2. Well, let's talk about phase 3. So phase 3 involves the elimination of estrogen from the body. So can you actually get rid of it? Dysbiosis, again, can impair phase 3. And that's because that beta-glucuronidase can interfere with phase 3. So if there is excessive microbe-secreted beta-glucuronidase, the estrogen goes back into circulation instead of being eliminated in the stool. So again, anytime you see issues with phase 1, phase 2, or phase 3 estrogen metabolism, think about the gut. It could be a factor. Now, we talked about estrogen. What about the thyroid? Let's talk about how thyroid dysfunction can be caused by the gut microbiome. Now, here's a couple great articles, one from 2020, one from 2019. The one to the left talks about how dysbiosis has not only been found in autoimmune thyroid disorders, but has also been reported in thyroid carcinoma. So autoimmune thyroid disease and thyroid cancer linked with gut dysbiosis. Additionally, the composition of the gut microbiota has an influence on the availability of essential micronutrients for the thyroid gland. Again, if the gut isn't healthy, you can't absorb these nutrients. So the thyroid gland, we talk about iodine, iron, copper, selenium, zinc. If those aren't right, you can't make thyroid hormone, you can't convert thyroid hormone. So we can run into issues again with the thyroid function if the gut is dysbiotic. Now, the study to the right talks about, again, that autoimmune link. And altered microbiota composition increases the prevalence of Hashimoto's thyroiditis and Graves' disease. Microbes influence thyroid hormone levels by regulating iodine uptake, degradation, and enterohepatic cycling. Now, here's one talking about hyperthyroidism. So studies have shown that hyperthyroid individuals seem to have a decrease of bifidobacterium and lactobacillus and an increase in enterococcus. So again, things we can look for on a stool test, identify, and correct. Now, here's another one talking about thyroid cancer and thyroid nodules. Again, certain organisms have been linked with these. These researchers found that in comparison to healthy controls, the relative abundance of Neisseria and Streptococcus was significantly higher for thyroid cancer and thyroid nodules. Butyrosomonas and lactobacillus displayed notably lower relative abundance for thyroid cancer and thyroid nodules, respectively. So again, so many different thyroid conditions can go back to dysbiosis in the gut. Now, it's not just dysbiosis, an imbalance of bacteria, but Pathogens can also affect the thyroid gland. So here's one on H. pylori and autoimmune thyroid diseases. Here's H. pylori infection prevalence was significantly increased in Graves' disease and Hashimoto's thyroiditis patients. So if I have a patient with Graves or Hashimoto's, you can bet I'm looking for H. pylori. Now here's one on blastocystis hominis, which is a parasite. Patients with Hashimoto's thyroiditis showed a significantly higher serum interleukin-17 compared with controls. Interleukin-17 was significantly higher in Hashimoto's patients infected with blastocystis hominis compared with Hashimoto's patients not infected with blastocystis hominis. And then they treated the blasto and saw what happened. After blastocystis hominis was eradicated, TSH, anti-TPO, and interleukin-17 were significantly decreased. So again, if I have a patient coming to me with Hashimoto's, I'm especially looking for H. pylori and blastocystis hominis. If I find them on the report, I'm addressing them. Now, let's also talk about the different ways, some other ways that the gut bacteria can affect thyroid function. Gut bacteria influence thyroid hormone conversion. We know that about 20% of thyroid hormone conversion takes place in the gut. The commensal gut bacteria, the good guys in the gut, facilitate this conversion of T4 to T3. So in the GI tract itself, T4, the inactive form, is converted into two forms of T3 sulfate, which is T3S, and triiodothyroacetic acid, which is T3AC. Now, the conversion of this T3S and T3AC into active T3 that we can utilize requires an enzyme called intestinal sulfatase, which is produced by commensal gut microbes. So if there's gut dysbiosis, we're going to have a problem. Gut dysbiosis significantly reduces the conversion of T3S and T3AC to T3. Now, LPS from those gram-negative bacteria can also get in the way. LPS can impair the conversion of T4 to T3 by inhibiting the 5-deiodinase enzyme, which converts T4 to T3. So dysbiosis negatively impacts thyroid function in many ways. So we have that LPS can decrease expression of thyroid receptors, reduce T4 and T3 levels, increase reverse T3 levels, and promote autoimmune thyroid disease. Now let's talk about some hormone-related conditions and the gut microbiome, starting with female reproductive tract disorders. So we have many different microbiomes. So there's the gut microbiome, but there's also vaginal microbiomes, uterine microbiomes, and studies have found that a dysbiosis pattern in the gut, the vaginal, and uterine microbiomes, that this can be associated with estrogen-driven disorders, including endometrial cancer, endometriosis, and uterine fibroids, and disorders associated with estrogen deficiency, such as PCOS. Now, the study to the right, which is from 2024, they say that the gut microbiome, considered an endocrine organ, plays a crucial role in the reproductive endocrine system by interacting with hormones like estrogen and androgens. Imbalances in the gut microbiome composition can lead to various diseases and conditions, including PCOS, endometriosis, and cancer. So let's talk about endometriosis. Now, just for the sake of time, I'm going to skim past a lot of this, but a lot of you are familiar with endometriosis and that it occurs when tissue similar to the lining of the uterus grows outside the uterus. And this tissue responds to estrogen and bleeds during menstruation, which can cause local inflammation and severe pain. Now, it is interesting that up to 90% of patients with endometriosis experience gut symptoms like bloating, nausea, vomiting, constipation, and diarrhea. So again, it seems like, is there a link with the gut? Now, the exact cause of endometriosis is still considered unknown. There are several theories, including retrograde menstruation, where the menstrual blood and tissue flows back through the fallopian tubes and into the pelvic cavity. The problem with this theory is that While 90% of women have retrograde menstruation, only 1 in 10 develop endometriosis. So, new research is suggesting that bacteria may play a role in endometriosis. And sure enough, studies are finding dysbiosis in the female reproductive tract and the gut microbiome has been associated with endometriosis. So here's a study from 2021, again, talking about that connection between the microbiota and endometriosis. Now, there's a great chart too coming from this, and it's talking about the beta-glucuronidase link again. So a lot of this can come back to that beta-glucuronidase imbalance. This talks about one way that the microbiota can be related to endometriosis is through the regulation of estrogen. When there is gut dysbiosis, microbial beta-glucuronidase secretion can be upregulated, which increases estrogen abundance. And it is suspected that the gut microbiotas of endometriosis patients have a larger number of beta-glucuronidase producing bacteria. And these bacteria that are capable of beta-glucuronidase production are often referred to as the estrobolome. Now, here's several studies linking different things related to the gut with endometriosis. So this first one is talking about the beta-glucuronidase link. And again, we have that link between the interaction between the gut microbiota and how that estrobolome has been regarded as one of the key factors of endometriosis by dysregulating that estrogen availability. But that's not the only thing. It's not just beta-glucuronidase that could lead to an endometriosis picture. LPS from the bacteria can also be problematic. Here's a study, again, 2024, new research coming out. LPS could be involved in pelvic inflammation and promote the toll-like receptor 4, NF-kappa B mediated growth of endometriosis. Now, here's another way, mast cells. There is evidence that mast cells play a role in endometriosis. And researchers are saying, this is a 2017 article, targeting mast cells might offer new alternatives to treat this disease. Researchers have found that the numbers of mast cells and activated mast cells were clearly increased in endometriotic lesions of both animals and humans. So again, on a stool test like the GI map, do we have high levels of these mast cell activating microbes? We might see an overgrowth of some of these opportunistic microbes here. So we're looking for that picture in our endometriosis patients. Here's some more talking about dysbiosis. Now, this is talking about dysbiosis in the cervical mucus and in the endometrial tissue. So we look at all these different microbiomes, but we do know that the gut microbiome can sometimes act as a reservoir for bacteria that can seed some of these other microbiomes. So we do have to look at the gut in all of this too. Now, some studies recently have focused in on a particular organism, Fusobacterium. This one study from 2023 found that 64% of patients with endometriosis, but less than 10% of controls had Fusobacterium infiltration in the endometrium. Now, this other study from 2023 as well found that, it appears that this Fusobacterium infection of the endometrium was responsible for the development of endometriosis. Fusobacterium is a common member of the oral and gastrointestinal tract microbiota. In fact, sometimes we see it in high amounts in the gut because it's in the mouth and we swallow it every day in the saliva. And if someone has low stomach acid, they don't kill those oral microbes very well. So if I'm seeing high levels of Fusobacterium in the stool, I'm instantly questioning, do we have a situation of low stomach acid here? Is this coming from the mouth? Now, this study also noted that Streptococcus, Proteus, Klebsiella, and Pseudomonas were highly detected in endometrial samples from women with endometriosis. So again, on the GI map, I'm looking for high Fusobacterium, but also high levels of some of these others. Again, we're talking about the endometrial tissue dysbiosis here, but could we also see this in the gut. So that's what in the stool we're looking if there's a gut connection to. Now, here's some again talking about the cervical microbiota, but also the gut microbiota. Now, this one was interesting that they were finding higher levels of some microbes and lower levels of other microbes. So when we're looking at the fecal microbiota, so these are the ones showing up in the stool, they found that 10 taxa, which if you look at the list of these organisms, these are butyrate producers. These 10 taxa, these butyrate producers, were significantly depleted, so lower levels of those. And 2 taxa were significantly enriched in the stool compared with those in the normal groups. So again, we're looking for, are there some low butyrate producers here? Is that a factor? Another one is looking at, there's in the peritoneal fluid, they're actually finding that there can be higher levels of Pseudomonas. So again, I do look at some things with the gut too, because we can measure Pseudomonas in the stool. So I'm looking to see if there's an imbalance with that Pseudomonas level. Now, it is interesting that, while some of these studies do look at the stool microbiota of women with endometriosis, while others are looking at endometrial tissue itself and the microbiome there. Some are looking at the cervical mucus microbiome. This study from 2023 concluded that we found the bacteria in the gut, as opposed to any other site, are required for endometriotic lesion progression. So again, we're looking at dysbiosis in any of these areas, but they're finding actually the gut seemed to be the key factor. Now let's talk about another condition, PCOS. Again, for the sake of time, we'll skip a lot of this information on what PCOS is, but we know that it's characterized by persistent anovulation, insulin resistance, and hyperandrogenism. But like endometriosis, the exact cause of PCOS is still unknown. But studies suggest, once again, dysbiosis of gut microbiota may play a pathogenic role in the development of PCOS. And there seem to be certain aspects of this that can be caused by different microbiota. And here's a nice chart for that study that's showing all the different factors that can lead to PCOS. And you'll notice that one of those here is gut dysbiosis. Again, a couple studies here. So what is this link? How is this gut dysbiosis leading to PCOS? Well, this study from 2023 says a large body of studies demonstrated that gut microbiota could regulate the synthesis and secretion of insulin and affect androgen metabolism and follicle development. Here's another one talking about, they looked at the microbiomes of PCOS women and found that as compared to the normal group, what they found in PCOS women is that they had lower alpha and beta diversities, as well as a decreased relative abundance of bifidobacterium. And then they had an increased relative abundance of Bacteroides, Parabacteroides, and Clostridium. So there seems to be this dysbiosis that can cause a disturbance in gut metabolites, with things like the short-chain fatty acids and bile acids, that again is going to lead to things that could create a PCOS situation. And again, they say that, the short-chain fatty acids, the bile acids, the branched-chain amino acids, all these metabolites can directly regulate the secretion and sensitivity of pancreatic insulin. So we always have to be thinking back to the gut in all these conditions. Here's another one. An altered gut microbiota significantly correlated with PCOS phenotype. Now, they're finding that the groups increased in PCOS include bacteria belonging to Bacteroides again, Escherichia shigella, and Streptococcus. These were negatively correlated with ghrelin and positively correlated with testosterone and BMI. Now, on the flip side, the ones that were decreased in PCOS included bacteria from Akkermansia and Ruminococcaceae. So these showed the opposite relationship with body weight, sex hormone, and gut-brain peptides. Now, bacterial LPS, again, can be a factor. They've shown that the LPS can induce chronic inflammation, obesity, and insulin resistance, and that can set the stage for the development of PCOS. What about uterine fibroids? Yeah, again, there's research connecting dysbiosis in the gut. So So this study from 2022 found that alpha diversity in patients with uterine fibroids was significantly lower than that of healthy controls. So there were alterations in multiple phyla, such as the Firmicutes, Proteobacteria, Actinobacteria, and Verrucomicrobia. So again, taken together, it is possible that gut microbiota dysbiosis has the potential as a risk factor for uterine fibroids. Now, this study to the right from 2023 talked about what are the different ways that this may happen? Well, a few ways by which the gut microbiota contributes to uterine fibroids include estrogen, impaired immune function, inflammation, and altered gut metabolites. Here's a nice little chart again from that article, the little flow chart showing how the dysbiosis can lead to uterine fibroids. What about unexplained infertility? Again, there's a great study here from 2022. So they found that the most striking phyla level change in the unexplained infertility group involved depletion of actinobacteria. Now, bifidobacterium is part of that actinobacteria and abundance of proteobacteria. Now, proteobacteria is a phylum that has a lot of pathogenic and pro-inflammatory organisms in it. So they're showing that this suggests a critical role of proteobacteria phyla in unexplained infertility. And it also concurs with the fact that bifidobacterium, which is a genus of the depleted phyla Actinobacteria, inhibits gut pathogens. So there's too many bad bugs, not enough good bugs here. So they found that the unexplained infertility group had the highest enrichment of pathogenic gram-negative families who have that LPS. In fact, the unexplained infertility group had almost twofold higher levels of proteobacteria than the controls. So again, the overload of these LPS-producing gram-negative bacteria activates the gut innate immune system, generating interleukin-6-driven systemic low-grade inflammation, ultimately leading to unexplained infertility. Reproductive cancers in the gut microbiome. Lots of research showing up here on breast, cervical, and ovarian cancer. So again, due to the sake of time, I'll kind of skim through this. But again, here's that gut microbial beta-glucuronidase link again, where they found that some species of the microbiota that produce the beta-glucuronidase were abundant in breast cancer patients. Again, it's going to lead to that higher estrogen level, higher estrogen circulating in the body. Here's some more, again, of how important that the microbiota play that important role in implications for breast cancer. Again, beta-glucuronidase could play a role, and not only for recirculating estrogen, but it also, that beta-glucuronidase also deconjugates endocrine disrupting chemicals, such as bisphenol A. So if you have high beta-glucuronidase, estrogen stays around in your system too long, but so do these toxic chemicals that have also been linked with cancer. So we have to think about that implication with high beta-glucuronidase. Now, we can't forget about the guys here. There's also a link between the gut microbiome and prostate cancer. And again, they found that certain organisms seem to play a bigger role than others. Here's one that says patients with prostate cancer had a higher ratio of Bacteroides and Streptococcus species in their gut microbiota. So some bacteria can affect testosterone levels through deglucuronidation of testosterone in the gut. And men with enlarged prostates, according to this study, had significantly higher Firmicutes to Bacteroides ratios in the gut microbiota. So again, we have to think about all of those implications here. Again, here's another one with a great graphic on the microbiome and prostate cancer. Now, this one talks about a higher relative abundance of a particular species of Bacteroides, but also they found that, in comparison to patients without prostate cancer, the prostate cancer patients had lower levels of Faecalibacterium prausnitzii. So they were lacking this major butyrate producer in the gut. So again, we want to look to see, do we have high Bacteroides, high Bacteroidetes? Do we have low Faecalibacterium prausnitzii? Now, this is bidirectional. So far we've talked about how the gut microbiota can affect the hormones. Well, it goes both ways. Hormones can affect the gut microbiota. So estrogen influences the composition and the diversity of the gut microbiota. Estrogens and progesterone promote increased microbial diversity, maintain the gut barrier. So they're protecting against leaky gut by upregulating these tight junction proteins. And they're protecting the gut from injury. And that's why in menopause, when estrogen levels start to fall, it's associated with a lower gut microbiome diversity. Multiple studies have found lower abundance of Firmicutes and Ruminococcus, and higher abundance of this Bacterosomonas, Doria, Prevotella, Cetarella, and Bacteroides in post versus premenopausal women, or in women with just lower estrogen. So studies seem to suggest that low estrogen leads to less diverse gut microbiome and leaky gut. But estrogen also affects bile production and secretion. Both low and high estrogen can cause decreased bile flow. And then thyroid, that can come into play here too. If someone is hypothyroid, that can cause decreased bile synthesis. Now, bile, of course, we need it to digest fat, but it has significant antimicrobial effects in the gut. So if bile production or secretion is impaired, bacteria can overgrow, creating gut dysbiosis. But estrogen does so much more in the gut too. It affects gut motility. It can inhibit colonic smooth muscle contractions, which means that if estrogen is too high, it can lead to constipation, and then chronic constipation can lead to gut dysbiosis. When things are moving very slowly, we can see higher Bacteroides, higher methanogens, and so forth. So studies are showing us that estrogen protects against leaky gut and inflammatory bowel disease. We think about estrogen receptors and tissues that estrogen receptors are found on. Well, sometimes we forget about the gut. There are estrogen receptors, predominantly estrogen receptor beta, in the colon. So you need sufficient estrogen to activate this estrogen receptor beta. And then the estrogen receptor beta, when it's stimulated, is going to help maintain the integrity of the tight junctions and the barrier function and suppress inflammatory signaling. So again, if there's low estrogen, there can be decreased estrogen receptor beta activation, which can lead to increased permeability, altered gut microbiota composition, and increased intestinal inflammation. Okay, so let's wrap up with a quick case study. So here we have a 38-year-old female. She's having painful, heavy periods. She's having PMS, some breast tenderness, some mood changes. She's noticed some mild acne, and her belly feels hard and painful after eating. So let's look at her GI map and see what we found. Well, there is a little bit of norovirus here. Norovirus is the leading cause of foodborne illness outbreaks in the United States. Now, we don't necessarily have to treat it when it's showing up because it is a self-limiting infection, but it is a clue. So I'm always factoring this in, even if I'm not doing a direct intervention, because this can be a clue of low stomach acid. Because if norovirus is ingested on the food, one of stomach acid's main jobs is to kill these microbes ingested on our food. So if she couldn't kill the norovirus, there's a question, did she not have sufficient acid in her stomach to kill it on the food as it passed through the stomach? So again, my brain is starting to think about, okay, I'm going to look for other clues of low stomach acid here because this is making me suspicious. So H. pylori is one of those main organisms I'm looking at when I'm thinking about stomach acid. And she did have H. pylori. Now, it's a moderate positive, so it is more of a clinical judgment call whether to address it. But if I see signs and symptoms that fit, or clues on the report, like a lot of bacterial overgrowth or yeast overgrowth, I figure that it's probably up to no good. It's probably neutralizing stomach acid. So I'm probably going to do an herbal protocol to address it. Now, H. pylori is notorious for suppressing stomach acid. So my question is now, thinking of page 1 in context of this page 2 H. pylori finding, was this a factor that made her more susceptible to picking up the norovirus? In my mind, this gives a little more weight to the H. pylori. Maybe this is a bigger factor. Plus, we do see clinically and in the research that acne seems to be connected with H. pylori, and she has acne. So again, could this H. pylori could be playing a role in her acne? Plus, her hard, painful belly after eating. This is suppressing her stomach acid. We often see, gas, bloating, belching, indigestion, abdominal pain, symptoms like that from H. pylori. So again, this may be something we want to address. Plus, as coming up on the next slide, we'll see a lot of bacterial overgrowth. And again, with H. pylori, knowing how it tends to suppress stomach acid, and that can lead to bacterial overgrowth. Again, I look at this in light of that dysbiosis to think, is this an upstream factor that I need to address? Now, first, let's look at the good guys, the commensal organisms. We see that there's a clear pattern of low normal, less than DL. This is an insufficiency dysbiosis. Overall, there's just a lack of good bugs. Now, these are our short chain fatty acid producers. So there's probably a deficit of short chain fatty acids like butyrate, acetate, propionate. And what that means is there's probably going to be increased colonic pH, which we know can lead to increased beta-glucuronidase activity. Now, looking at the next page, again, there's some dysbiosis. There's bacterial overgrowth and yeast overgrowth. Again, we know that H. pylori can suppress stomach acid. That can allow things like staphylococcus that are swallowed from the mouth to overgrow. Candida likes that higher colonic pH, so that can overgrow. So again, is the H. pylori a factor? And we want to address the dysbiosis and that H. pylori. Now, this particular pattern is fitting a digestive dysfunction dysbiosis pattern. So again, we're looking at things like, is low stomach acid contributing? Is low enzymes a factor? Now, there's also an inflammatory dysbiosis pattern. Klebsiella is an LPS producer. Proteus is an LPS producer. Klebsiella is a super producer of histamine. So all these things that we talked about throughout this webinar. No parasites, fortunately, so we don't have to worry about those. Now, looking at her intestinal health markers, low pancreatic enzymes. She's not digesting her food properly, which means there could be more food for the yeast and these opportunistic bacteria. And there's that high beta-glucuronidase activity. So, the painful periods, the PMS symptoms, I'm highly suspicious now that this could be a factor. But now from everything else we saw before this, we know a little bit more why this beta-glucuronidase activity may be elevated. She also has high secretory IgA. Now, this is a proper, appropriate response to an acute norovirus infection. So I don't have an issue with that. Her immune system works. It saw the norovirus and it responded. So these are some interventions I would consider. Pancreatic enzyme supplement. We got to get her digesting her food better. And that could be helping too with that hard belly and discomfort after meals. I'm going to think about calcium D-glucarate for that high beta-glucuronidase activity. Now, realize that calcium D-glucarate doesn't fix the problem. It's just going to prevent the beta-glucuronidase from interfering. So it's going to allow the estrogen to exit the body, but it is more of a Band-Aid approach. So I'm going to use it so she has some symptom relief. So maybe she doesn't have the painful periods. She doesn't have the PMS because we're going to excrete that estrogen properly while I'm working on those root causes, the lack of the commensal bacteria, the high LPS producers, and everything else. So you can't just give calcium D-glucarate. It's not going to fix the problem. You got to work on these underlying things, but this can be great for symptomatic relief. Now I'm going to work on the H. pylori and do a natural protocol for that. I'm going to do a dysbiosis protocol with some great antimicrobial herbs. And then I'm going to support the low commensals. Now, commensal bacteria are fiber lovers, so we need more dietary fiber. Now, I may also bring in a prebiotic powder because these things were so low. Diet alone it's not going to move the needle as fast. So if we can bring in a concentrated prebiotic powder, it's going to help just speed things along. And I would also think about a probiotic with some lactobacillus and bifido, because those levels were low. So we can instantly just add more in the system, have them get to work right off the bat by giving them as a probiotic. So in summary, the gut microbiota may influence sex hormone levels through a number of mechanisms. High beta-glucuronidase activity can cause estrogen dominance and increase the risk of estrogen-driven conditions, including cancer. So always investigate the possibility of gut dysbiosis in patients with hormone dysfunction. Fixing the gut can correct hormone imbalances. And don't forget, there is a bidirectional relationship between gut microbes and hormones. Now, for more information, if you want to learn more about the GI map test, which was featured in this webinar, you can go to the diagnosticsolutionsLab.com website. You can download the interpretive guide. If you need help in interpreting the report, we are available for clinical consultations. And then I have my own consulting business, Pomeroy Institute. I do one-on-one practitioner mentoring sessions, and I have some comprehensive practitioner training courses. And in conjunction with this webinar, I am offering a couple coupon codes. So you can save on my GI map interpretation course and my Dutch interpretation course with these codes that are on the slide.
Welcome to Rupa Health, the best place to order, manage, and track results from over 30 different lab companies in one single place for free. It's going to take you under 2 minutes to sign up, and you can order any functional medicine lab for your client in under 30 seconds. Let me show you how it's done. So here's our beautiful interface. I'm typing in the name of my client, selecting the lab that I want to order for them, and hitting send. From there, Rupa and their amazing team handle the rest. They email the client, collect payment, and even offer an interest-free 3-month payment plan. We've also built the world's largest library of information about chronic health conditions, the lab tests that can help you find the root cause, and the evidence-based interventions that you can use to help people heal from them. It's called the Rupa Health Magazine. There, we have in-depth articles about almost any health condition you can imagine. And we give you step-by-step protocols that other clinicians have used to help their clients heal and that are verified by evidence-based sources. You should totally check it out, and it'll transform your practice. And we can't wait to see you. So make sure you sign up today at rupahealth.com.